Okay. So hi, everybody. It's a great pleasure to have with us Merav Achisar from the Hebrew University today. She's a professor in psychology and brain research at the LSEC in the Hebrew University. She's interested in learning, perception, and skill acquisition, both successful and less successful, which I think is a very interesting combination. Her education, very briefly, BA in biology and computer science, a master's in physiology, and then a PhD in neurobiology with Shaul Ochstein, all in the Hebrew University. Then she went to San Francisco to do her postdoc with the famous Mike Merzenich. I don't know where is he today, but big name. And Merav has been highly influential in all these domains that she studies, and been gravitating, in my eyes at least, more and more towards the clinical arena. So we're very happy to hear today about autism and dyslexia, failures in acquiring specific skills for opposite reasons. Thank you, Merav. The floor is yours.
The floor, okay. So thank you, Moshe. It's a pleasure. Initially, when we talked about me giving a talk, it was about giving a talk in person. And I actually really like the idea that it's going to be a talk in person. And why do I prefer talk in person, even though it's so much easier not to travel to Bar Ilan and stay in my office, etc. So it's so much more convenient because a talk in person is more interactive. I can get a feel for what people are doing, for when they get bored, for when they get tired, and then change my long-term plans of how to give the talk and perhaps stop because it's so boring or whatever. Or anyhow, try to update the pre-plan for what I'm going to say, taking into consideration the online sensory cues and affecting my sensory motor plans, even though nothing was explicitly said. And this point is a crucial aspect of the content of my talk, as you may guess, which is associated with the autism aspect of my presentation. And following what Moshe said, I'm not into clinical aspect. I'm much more, in a way, pretentious than that. I'm trying to explain clinical phenomena or clinical manifestations with, to the best of my ability, a single unifying underlying account, which obviously doesn't explain everything and not about everybody, etc. But I'm trying to see how much we can take a very simple, basic idea. So let me, why did I, sorry. Okay, so the two developmental difficulties whose prevalence is growing and growing, probably if we wait a bit, will be very, very large portion as it's defined in the population, are autism and dyslexia. So what do we know about autism beyond being really, at least in terms of diagnostics, becoming more and more prevalent to the extent that now it's evaluated in more than 1% of the males and about a quarter of that in females in the population. So we all know that it's associated with social deficits, difficulties in communication, and I'm referring to high functional people with autism, namely they don't have a language difficulty, but still they suffer great difficulty in understanding social situations. It doesn't mean that they don't care about them, but they don't feel for what's going on exactly, particularly when it's not expressed explicitly. But this is not the only characteristic, and these other aspects have recently been part of the formal diagnostics. And this also includes stereotypic and repetitive behaviors and kind of limited interests, and some aspects which are still in dispute of atypical sensory sensing. As we said, we study individuals with no language difficulties. We also assess their reading skills, and they're pretty similar to that of our controls. And most of our controls are kind of adults, student-like population, either now being students or were or will be students. And how is it related to dyslexia? Well, developmental dyslexia, as we all know, it's associated with consistent difficulties in becoming proficient readers. It's not about the comprehension of the written text, it's about deciphering in a very fluent way the written script, typically the alphabet. Whether you become an expert means that you can quickly read, and reading is kind of a platform for acquiring information, and you don't have to struggle with the process itself. Both of these uh, clinical evaluations, I'm thinking about difficulties in acquiring skills. In one, the skill of being an expert. In one, being an expert is being a very, very, let's say, fluent reader. This is difficulty of dyslexia. And in autism, I define their difficulty as becoming social experts. So experts in the culture, in the habits, in the non-explicitly said habits of social interactions. By the way, these habits differ between populations. I could be within a population whose language I speak and still feel very uncomfortable because I'm not familiar with the all sorts of social informal or non-explicit agreement. And both of these developmental difficulties, developmental atypicalities are rather specific. And when people say specific, it doesn't mean that this is the only difficulty they have, but rather, as we do in the lab, that it's not a general reasoning disability. So we can compare the other abilities, reasoning scores, and sort of in formal evaluations, let's say AQ-like or IQ-like and reasoning abilities. So these are matched across the groups that I'm referring to, controls or typical population, people with dyslexia and people with autism. So I'm referring to, okay, given that they're matched with age and matched with reasoning skill, what's going on? For that, let me talk just a bit about what is perception, because intuitively we think of perception, well, this is our immediate experiences, but it's important that this is our brain's immediate interpretation of external stimuli. So we are sort of bombarded with stimuli in most environments. Typically, these are environments we're used to. We don't have to elaborate but you have an immediate perception of what's going on. But we need to remember that this perception is an interpretation. And this interpretation takes into account both the responses to the sensory stimuli, what we are stimulated with at this very moment, but also what we expected to be stimulated with. And when I say expected, it doesn't mean that I can explicitly say it, but that all our system is kind of accumulating information. This is what experience is about, accumulating information and kind of having predictions based on these experiences as to what will be our next stimuli. Let me give you an example. So whether you identify this picture or not depends on your personal experience, on your sensory experiences. And if you're a psychology student, you probably have seen it. If you haven't, it's novel to you, and you will have difficulties in identifying what this picture is about. So this is one picture, and this is another picture. Well, this is kind of an impoverished, the previous one was an impoverished picture of this, which is more clear. We can all see what it is. Now the question is, how will this experience affect our perception when we are exposed again to the previous picture? Okay, I will go back to the previous picture, and hopefully now we can clearly perceive that it's a cow. And the reason I'm showing it to you is to show you fast effects of recent experience on current perception. Okay, so what I hope you experienced is that initially this was not very clear, and then you had this experience, and then you could clearly see that this is a cow. But the point is, it's not about memory, explicit memory, but you can clearly perceive it's a cow, right? So this was a recent effect of previous stimuli on your perception. And this is not just about visual stimuli, it applies also to auditory stimuli. I hope it works. So let me sound it out. This is the auditory equivalent of the unclear cow. Okay, I suppose you couldn't clearly perceive what was said, but then I will show you the clear example. Oh, I tried to. Oh.
The man's painting a sign.
I hope you can all hear it, right? Okay. Just again.
The man's painting a sign.
Now let me go back to this one. Okay. I hope you all had a clear experience of clear perception. So this was fast plasticity in a way, because you had the same experience, then you had another one, and then the previous sensory stimulation had a different perception, yielded a different perception. This is what I call effect of recent stimulation. But we're also, of course, affected by a cumulative stimulation. I could bring an example for that. I don't think I brought an example for that. But basically, there's lots of experiences that we have had throughout our lives, or for hours, or for days, or for at least years, and they affect the way we perceive stuff. By the way, a very clear example that I can just tell you, and you can, well, is how we hear language, right? We, people with a Chinese background will not decipher between la and ra, right? It's obvious, even though we do. It is also implemented, is also manifested in the way they say la and ra. They're not so different. Why is it? Because they had these experiences where the point of information, the place where this the discrimination resides in the stimulus is not attended, it's not informative when you speak Chinese, because these are not different phonemes. So it's kind of an example for an accumulative effect. So let me just say, I talked a bit about autism and dyslexia and a bit about perception and how we accumulate information which affects our prediction, and information is accumulated long-term and short-term. What I'd like to convince you with in the rest, which is kind of probably most of the talk, is that these two windows of learning, of in a way learning the statistics of the environment, are differently related to different skills. I'd like to convince you in the remaining part of the talk that acquiring proficiency in both in language, in first language, second language, and particularly reading, is based on adequate familiarity with long-term statistics, whereas acquiring proficiency in interactions, in social interactions, is based on recent statistics. So what is recent statistics? This was just in a way the example I gave you about the difference between giving a talk in Zoom and giving a talk in person. So there's something ongoing which has to be updated by recent cues. I'm looking now, rather than looking at my presentation, I'm looking at Michal's face in a way, and I'm basing my rate of conversation on Michal's be aware, big responsibility on Michal's online responses, because these responses are important for me to get a feeling for what's going on. Had I not been seeing anybody, I would have continued with my routine, with my long-term based routine, and it would not have been affected by ongoing interaction. What does such interaction require? My ability to integrate these nonverbal cues and update my motor plans, my all sorts of plans online, given this information. What I'd like to convince you is that this kind of fast update is impaired in individuals identified with autism. What I'm going to say is that this difficulty is not specific to social interaction. I will show you this difficulties in situations which are lab determined. There's no other people within this task, but will be characterized with slow update of both predictions, sensory predictions, and motor plans. And so in a way say that the social difficulty is not the source, but is an outcome of difficulties in fast updating, which is mostly manifested, but not only, also in sensory motor interactions, mostly manifested in social environments. It doesn't mean
excuse me just for a second. People are complaining that they can't hear you well. I wonder if you're using your headphones, or what do you do there for?
Can you hear me well? Can you hear me better now? I just got closer.
I think it's much better, yeah.
I wonder, I'm not going to say everything again, right?
No, but they got the low spatial frequency so far.
Okay.
You don't have to repeat, but if anybody still doesn't hear Merav well, just let us know. Otherwise, we can just continue.
So if it's still a problem, please let me know. Michal says it's okay.
Okay, yeah.
I just got closer. So I'm not with the headphones, but I just got closer to the screen.
Thanks.
I have a crappy microphone, right?
Thank you.
So anyhow, what I was saying, just to summarize, is that what I will try to show you is regarding autism, that it's a slow update, which is mainly important for social interactions, but it's not the reason. Social lack of interest is not, I don't think, is the original reason. It doesn't mean that it doesn't have an effect too. Once you're not good in social interactions, you may be reluctant to continue. But basically, I will show that the slow update is not necessarily only in social interactions. And what about language and reading difficulties? I will try to suggest that fast, fluent deciphering of the written script is impaired by the opposite atypicality in learning the statistics, meaning a difficulty in long-term accumulation of the task-relevant statistics. For example, in one's native language, you have the statistics of the syllables, you have the statistics of the word, of specific phonemes. And if you have that, even though you're not aware of it explicitly, you will have these proper predictions, which will make the reading and, of course, also understanding process much faster and much more fluent. So I tried to show you this dissociation between different types of difficulties in statistical learning, which are manifested in these two difficulties of skill acquisition. Can I be heard sufficiently well? Okay. If I say it again, you will have easier time to understand me because you have hopefully some predictions regarding what I'm saying. So all that said, the task that we're actually using, well, I'll show you more than one task, but the task that has been our sort of home task has been this one. So we serially present two tones. This is it, but we have many steps. We have hundreds of steps, hundreds of tries, and in each try, the participant is asked to say which tone was higher. And our intuition, and this was historical intuition, is that this is a simple, low-level perceptual task because it's all about simple tones, frequencies, and frequencies are deciphered already at the level of the inner ear because we map complex sounds to their frequencies. Well, what we are really interested in is how performance in this task is affected by the person's history. And for that, the fact that these are very simple sounds, these are pure tones, is an advantage for us for two reasons. One, because these are not sounds that people are familiar with. They don't have really long-term history with them because we are in the actual environment. We are confronted with very complex sounds. We are confronted with speech sounds, not with pure tones. So in a way, I'm the master of the statistics that individuals are exposed to in this task. So long-term effects would be 15 minutes ago, recent would be 2 and 3 seconds ago. So I'm referring to the type of statistics that I give participants in the task. Second is because it's a simple task, I can analyze it, and I won't get to the technicalities of how we do that, but we can analyze the effect on participants' judgment of the recent stimuli, pum, 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 in the T-1 trial, T-2 of the very recent stimuli versus the effect of all the other stimuli, let's say the 100 other stimuli that I gave people. And what happens with people is that their judgment is constructed implicitly. They're unaware of it. I am also unaware of this process when I perform this task, even though I'm very, very experienced with it. Their judgment is constructed to previous stimuli. Namely, by the time I hear the second tone in each trial, which is about a second after the first tone, my memory of the first tone is more similar to previous tones that were presented than to the actual tone that people heard. To what extent it differs across individuals, I will not get to it, but it is affected by previous stimuli. So the question is, how is it affected? Is it affected more by more recent stimuli, by everything? And I would really like to ask you, what do you think would be a good policy for the system? What should be more effective, more recent stimuli or earlier stimuli? Obviously, if I'm not a very good perceiver, some information, some cues from the past could be beneficial, but from which past? Very recent one or earlier ones? Well, it's unfair that I see Michal and Moshe, but I can see Roy as well. Yeah, Moshe, what do you think? Oh.
I think the more recent one.
So, well, okay, it's a trick question. I like trick questions. I mean, I like asking them. I'm not sure I like being asked with these questions. So it depends. What does it depend on? On the nature of the environment. So if the environment changes, if the statistics of the environment changes, pom, 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 I want to address recent stimuli because all earlier ones will lead me to expect wrong stimuli. On the other hand, if the environment is very stable, pum, 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 and it doesn't change, I want to have large average because my samples are noisy. And the more samples I have, the better my estimation is. We know we teach it in statistics. It goes by the square root of the number of samples. Our evaluation of the mean improves at this rate. So if the environment is stable, we want to have long range statistics. If it changes, we want to address recent ones. How do we know? Can we change it? So, well, actually, it's pretty difficult to know because even when the statistics is stable, there's some variability. So how can you tell if it's slightly higher, whether now statistic has changed, or it is within the range of variability? And what happens with the system? So we can now, I mean, it's not that difficult, but we now can evaluate what is the effect. So this is it doesn't matter. The units don't matter. What is the weighting that our judgment puts on most recent? The most recent trial, -1, T -1, earlier one, and 2 or 3 trials earlier. And we can see that our highest weight here within these 3 trials is on the most recent one. This allows us some flexibility. If statistic changes, we'll take into account the most recent one more than others. But what about the all other ones? Well, per trial, our weighting decreases, but if we accumulate the information or the weight of all previous ones, we call it infinity. Our infinity is not very large. All the trials that we have presented, these also together have a substantial impact on our weight. So what I'm showing here is that there's a recency effect. We take into consideration implicitly the most recent events and the earlier ones. So this allows us to be prepared or to take into consideration when the environment changes, be prepared, and also have some stability based on repeated information. So now this is how controls let's say how control participants behave. What do we expect from participants with dyslexia? So if participants with dyslexia, as I tried to introduce, have a general difficulty in retaining information and getting the long-term statistics. How did I allude to it? Well, let's first, well, I already said that. So this is, let's see the results and then I'll unpack what I think about them. So in blue is our controls, in red is our dyslexic participants, and this is the weighting of the T -1, the most earlier one, the most recent one, second, 2 earlier, 3 earlier, 4 earlier, and this is the total. So the weight of overall stimuli that were presented throughout the experiment is smaller in dyslexia. How do we think it's related to their reading difficulties? We think that it reflects a general difficulty in retaining the perceptual trace. So what happens is that we have a long perceptual trace. It affects our decisions. As I said, we are biased. Here we measure our bias towards earlier trials. And the most recent trials are similarly affecting dyslexic decisions. However, altogether, the accumulation of the statistics is reduced, at least to the extent that is manifested in their decisions. So what do we expect that faster decay will do? We think that faster decay of this information will lead to slower long-term accumulation of statistics. something like a leaky capacitator. So it's like, yes, there is information that is added, but at a slower rate. So suppose their learning, long-term learning of statistics is slower. Before that, let me show that this is also the case. Here is Sagi. So Sagi is also here. It's not only in Tel Aviv. Just as we're all here, Sagi is also here. It's all virtual anyhow. So this is just a similar experiment that Sagi did to show the dynamics of implicit memory among dyslexics in the context of reading. This is just similar in a way to the two-tone, but here it's in the context of reading. As you can see, these are non-words, and participants were asked to read them. They are serially presented. Once you read a word, the second one, the next one will be presented, etc., etc. And when you see here, you see a repetition. When the word is repeated, reading rate is faster. The delay before reading becomes shorter. For how long? Well, if it's repeated like Klusko here, with a longer inter-word interval, the benefit is smaller. Meaning our interpretation is that the perceptual trace somewhat decayed and the benefit is smaller. Now we can track these dynamics both in dyslexia and in controls. And what we see is when the interval between the first and second repetition is 2, 3, 4, something like that seconds, the benefit for two populations is very similar. It's pretty large. It's more than 150 milliseconds faster in the second reading than in the first. However, if we wait a longer interval, well, in both groups, there is decay of the benefit, but in the dyslexic group, the decay is larger. So we interpret that both of these observations as a faster decay of the memory trace within seconds, which also affects long-term accumulation or accumulation rate when you're encountered with the same stimuli for many, many years. So how can we look at what happens across many years? We can't really, I mean, we could, but we don't. Perhaps we could, but we can look at common stimuli versus non-frequent stimuli, okay, in the language and see the comparison. And what happens is that we have, I think, a non-intuitive prediction. Let me go through this prediction. It's seemingly simple, but it's somewhat counterintuitive. So what is the intuition? What is the prediction that we have? So this is the exposure. So this is the rate of learning. In a way, this is amount of exposure, and this is how you perform as a function of exposure. So the more you're exposed to something, let's say syllables, your first language, reading, words, whatever, you improve. And this applies to both controls and to people with dyslexia. However, our claim is that people with dyslexia, the rate of learning the statistics, the overall rate is slower. So they would have a shallower slope of learning. So far, I think people will agree. However, it yields a non-intuitive prediction. And the non-intuitive prediction is that the group difference will increase with exposure rather than decrease. This is non-intuitive to most people who study dyslexia because there is a notion that with the years, people with dyslexia catch up. We're saying something the opposite. How do I interpret the notion of catch up? Because most tasks have a ceiling effect. That is, if you say, okay, this is good enough performance, okay, let's say this is 100% performance in a task, then with time, people with dyslexia will reach this level. So in order to test this prediction, we need tasks that are not saturating with experience. Now, learning does not saturate. This is well known in the literature of learning across the board. There is what is called the ubiquitous power law, that you keep improving even after zillions and zillions of exposures. And this is also the case here. So interestingly, one task that does not saturate is our memory span. If you look at people's memory span, it increases for more frequent words or to words that this person was more exposed to. And one such case, and this is the case that Eva used. Eva is collaborating in Haifa these days, but she's also here now, as you can see. So Eva said, okay, I will look at the span of digits, but I will look at the span of digits both in Hebrew and in English. So both have exactly the same semantic prevalence because it's the same word. But of course, people, Israeli people, or Hebrew speakers, they know English words. Of course, we all know English digits. But when we are asked for span, so how many digits we can remember when we are presented in a sequence, the sequence is longer for Hebrew speakers in Hebrew compared with English. This is not surprising. Now the question is, what do we predict for people with dyslexia? And in a way, counterintuitively, we predict that for people with dyslexia, the larger difficulty will be for Hebrew rather than for English, because with Hebrew, they have more experience. Everybody has more experience, but they will gain less from the same amount of experience. Okay? And this is really what we found. In this case, there's hardly any difference between the spans in Hebrew and in English for people with dyslexia. This is in a way unfair. Why is it unfair to compare these numbers? Because spans in Hebrew are slightly shorter. Why are they slightly shorter? Probably because our words for digits are longer. They're two syllabics, echad, shta'im, shalosh, rather than 1, 2, 3. So it's unfair. So relatively, it's better in Hebrew, but still the difference is larger here than here. So even though we're looking, and this is kind of very counterintuitive because we know that people with dyslexia have difficulty in a foreign language. So it doesn't mean that they're very good speakers, but the main problem is with becoming experts, okay? They will not gain to the same amount with the same level of exposure. So this is just to summarize what I said about dyslexia. I try to explain dyslexia. I don't think it explains everything, but it goes a long way. In a way, it suggests that the well-known observation that people with dyslexia have shorter spans reflects difficulties in long-term memory rather than in short-term memory, because we always assess short-term memory with known elements. So we suggest that the difficulty is in the benefit from long-term exposure rather than from shorter exposure. And of course, it doesn't mean directly an impact on comprehension, but if it takes you longer to read, you divert some of your attention to the things that you wouldn't like, and this is the act of reading. And what about autism? What do we expect will be the case? I can't resist myself. I have to do this thumb. What do we expect will be the impact of previous history in autism? In a way, just the opposite. So this is already moving quick. This is the example with dyslexia that I gave you. And this is, so ASD, autism spectrum disorder, we consistently denote in green, and this is again controls performing exactly the same task. Now here, this is T-1. This is the effect of the most recent trial. It was larger in controls than in individuals with autism. However, when we look at the long-term effect, it's the same in both populations. So this is the observation. What I made of it, or my interpretation, is that why don't they take into account, or hardly take into account, this is almost zero, hardly take into account the most recent event, because they did not have enough time to update their predictions for the incoming stimuli. So they are slow in updating their predictions, their sensory predictions, and they base their predictions. It's not that they don't have, but they base their predictions in the long-term accumulation, on the long-term accumulation of statistics. That is suggesting that if they have enough time to practice, enough observations or enough listening, they will form adequate predictions. However, if there's changes, if environment changes, if there's things that are not predictable and should be taken into account, they will do it in a less successful way because they rely more on earlier events. I don't know why it doesn't. Okay. Sorry. Okay. So this is what I try to say, that we predicted that there are slow online updaters. It's relevant both in communication, sort of verbal communication, and in this context that has no social or no direct social aspect. Now we try to ask whether this also applies to modal plans. So suppose, what are modal plans? So we use this task. This task is that. So this task was originally introduced into the lab by Nori. Nori actually is physically now, I think, in Israel, but generally is in Germany, where he is in a lab. He's leading a lab in Frankfurt. And Nori is a master on, is a musician, as a master on tapping. Please tap with the metronome now. With your finger. Michelle, you're using your finger for the wrong thing. Let me do that again, so Moshe will have an opportunity to try to synchronize with the metronome. So Nori started, oh, sorry. Yeah, you don't have to tap anymore. Why is synchronization interesting? Sorry, let me try. Okay, why is it? So Nori started the project and Gal led it to its successful publication. So why is it interesting to do synchronization? Well, both in autism, but here our focus was on autism, and we use people with dyslexia as a control group, as a specific control group, because the point is that people with autism have difficulty in synchronization. Recently, there's lots of work on the role of synchronization in social bonding. My intuition is like I'm thinking about singing or about moving together with music, like dancing together with music. And I do think that implicitly I have some sort of shared bonding with people that dance with me. But there are more systematic evaluations of the role of synchronicity in social bonding. It's become pretty, common. And also there's several studies that show that when you actively synchronize individuals, even when they're very young, like moving swings together in synchrony, afterwards, when they're asked to perform a task that requires some cooperation, they're more successful than when the swing goes like in opposite direction rather than in synchrony. So it seems that synchrony plays an important role in binding, in social binding. And then we looked, okay, let's take the synchrony rather than online with other people to the lab to do tapping with the metronome that is not affected by your response. And there we can sort of model it, do something very systematic, and look at the following stuff. So this, let me just describe the basic task. This is the metronome time. This is time. This is the tap, the metronome beats, and you could see that the intervals change at some point, they increase, then they decrease. And this is where participants tap. Typically, participants tap slightly before the metronome. This is something very consistent. It's called, there's a synchrony. It's not synchronous. We perceive it's asynchronous. It's not. It's a negative asynchrony. So this is negative because we tend to press slightly before the metronome. So the mean asynchrony is negative. And what we're interested in is first mean asynchrony, but more importantly, is how reliable we are. So if we perceive this as synchronous, how consistent we are with respect to our 0 point, with respect to our mean asynchrony. And individuals with autism are very variable around this point. Individuals with dyslexia do not differ from the control population in this task. They're very, very similar. But what we really wanted to ask in this very artificial context is, what is it that limits people with autism in synchronization with the metronome? So for that, we used a model that was somewhat developed by Nori. And this model, the details are not important. What is important is that the model attributes the timing of the tap to 3 basic parameters. One is our ability to keep an interval, timekeeper Timekeeper is noise is how reliable we are in keeping a fixed interval. Our interval typically is 500 milliseconds. I can tap. People can usually tap even without the metronome. They can tap pretty reliably at a fixed rate. How fixed? Well, depending on your internal noise. So one aspect is how reliably you can keep a fixed interval. Another aspect is how reliably you can really press your finger at a given point in time. But both of them have some noise, and the question is how reliably you can correct your noise by correcting the errors. How do we know the errors? The error is the time difference between the difference between our min asynchrony and our specific tapping point. So the question we ask is, how fast can you correct the errors because of your noise? And this is called phase correction. If you do full correction, it means that this alpha, this parameter, which is the phase correction, is 1. You took the entire error, which is when you tap versus when you were expected to tap, and you corrected it to its full extent. If you corrected it only partially, the alpha is small. Nobody has an alpha of 1, nobody corrects everything, but what we specifically predicted is that the ability to retain an interval, a fixed temporal interval, would not differ between participants, and neither would be the actual motor command. So these are kind of specific. Each symbol is one participant. These are all the controls. These are all the people with dyslexia performing the same task, individuals with autism. And we see that the ability, this is the level of noise, the higher it is, the better, the worse it is. The ability to keep a stable interval does not differ between these groups. Neither does the ability to reliably give motor commands. What is really very different between the groups is the ability to online correct the error. So alpha is smaller, meaning they corrected a smaller portion of the total error as they perceive it. And this is what we find very systematically. It's not true for all of them, but it's, I mean, here these participants are pretty good correctors, but it's true for the vast majority of our participants, okay? So we interpret both the tapping and the seemingly very, very different difficulty or atypicality that they have in tone discrimination is coming from the same central deficit, which is, or central atypicality, which is slow updating of both your perceptual predictions and your motor commands when this is required online at the scale of half to 1 second to 2 seconds, at the scale which is relevant for social online interactions. This is also the case when the beat is changing. It's just that remember, dun, 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 right? The beat became slower. And so these are the 3 groups, the controls, the dyslexic participants, and the participants with autism. It's 3 different changes in the beat, and they track slower the changes in the beat. They're slower to incorporate these changes. So this is roughly what I wanted to say. This is where we are. We have some others, but this is roughly where we are in terms of studying these 2 groups behaviorally. One last thing I'd like to mention is, can we say anything about the neural basis of these difficulties? And initially, our candidate for difficulties in implicit memory, either that it is lasts for a shorter time window or that it takes longer to modify, is implicit adaptation. Why do we associate implicit adaptation with these statistical learning? Actually, we're not the first, not in the context of populations, but in the context of general population, specific population. In the context of general population, it has been shown that the dynamics, the personal dynamics of adaptation goes along with the dynamics of what I showed you in the contraction bias in perception. For example, if 2 consecutive trials are 15 seconds apart, you will not have this contraction. So the dynamics of this behavioral memory effect follows the well-known dynamics that people have studied before of tones in the auditory cortex, suggesting that that might be related. So we thought we'd assess the dynamics of adaptation, both in dyslexia and in autism. And in autism, we thought that if they forget faster, this is what we suggest, that the perceptual trace decays faster, it means that the duration of the adaptation, which is an indication of how long the implicit memory is, will be shorter. So we try to assess the duration of adaptation in dyslexia and also the duration of adaptation in autism. In the case of autism, we thought the duration of adaptation is longer and perhaps takes a longer time also to kick in. So how do we measure this? The duration, the dynamics of adaptation, how do I, okay. So here comes Sagi again. This is a study of Sagi, and Sagi did the following experiment. So he did this pum-pum, pum-pum. However, he did it in blocks. And every trial in all 4 blocks was the same, but what differed is the interval between consecutive trials. He had short interval, longer, longer, and longer, 9 seconds interval. The intervals were chosen based on the literature of adaptation. So we predicted, let's say from this one, if there's a trial, there's 9-second separation before the next trial, the first tone in the next trial will not be adapted anymore, even in the control population. But if we look at the 3-second interval, the next tone will be adapted, meaning the response will be smaller. Whereas in dyslexia, we predicted the response will be maximal after a shorter time interval. Okay, it's a bit of a, it's not that complex, but it's kind of complex for working memory. So here's the results. It's complex a little bit. I will just take you through it. I have this slide and another one, and I will complete that. So these are the responses. The first tone, this is the first tone. This is the second tone. In this case, they're 600 milliseconds apart. This is the response from the mean electrode, which measures the roughly auditory cortices. This response is called M for -1. This is P for +2, 200 milliseconds. And this is the response to the second tone. This will not be relevant for what I want to say. But the response to this tone comes after either 9 seconds or 6 seconds or 3 or 1.5, depending on the block. We are waiting for you. Okay, so this is the response after 1.5 seconds. This is the response after 3. This is after 6, and this is after 9. Response become larger because it's not as adapted. When you are far enough, you're not as adapted, so you're out of adaptation. We would have liked, in retrospect, to have something here to see that it doesn't increase anymore, but the major increase is here, okay? The major increase in response is between 3, this is just an increase of this, between 3 and 6 seconds interval, okay? Meaning with 9, there's another increase, but we predict that based on literature that if we had 10 or 11 seconds interval, the response would already be similar. This seems perplexing.
So the reason I'm confused, Merav, is that you're showing all these effects on the responses to the first tone.
Right, because the first tone, the second tone is always after 600 milliseconds. The structure of each trial is exactly the same. What differs is the interval from the previous trial, and the first tone let's say in the 9-second interval, this is what the one comes after 9 seconds, right? This first tone after the 6 comes after 6 seconds, etc. This always comes after 600 milliseconds. So for the purpose of assessing the dynamics of adaptation, it's less relevant. Okay, and we do the same experiment with dyslexia. And here what we find is that the big jump is not between 3 and 6, but between 1.5 and 3. The big jump in response, namely that the largest, that the duration of adaptation is shorter because by 3 seconds, you're already halfway through maximal response. Okay, so let me just summarize. This is something I'm still learning to clearly explain. I think I was worse before, and probably I could get better. So just to summarize, what we see is that the duration of auditory adaptation is shorter in dyslexia, namely already after a shorter interval, response is, let's say, almost maximum. Can we really show that it's in the auditory cortex? This is our most recent, publication. This is by Ayelet, and this has actually been published this week. And Ayelet has done a similar experiment in the magnet to see whether indeed people with dyslexia, again in red, are less sensitive to simple regularities. And these are not regularities that you can detect with 2 seconds. You need to have some repetitions in order to detect these regularities. And what we see here is that every symbol is a different person, is that people with dyslexia do not differ in their auditory response, both in the primary auditory cortex and in both sides and in the associative cortices. They're not sensitive to regularities. Their responses with and without regularities do not differ, whereas people without dyslexia respond more, have a larger response when there's no regularity. It's about the same range of stimuli. So what I want to say here is the bottom line is that duration of adaptation is shorter, it's longer in controls, and this yields slower accumulation of long-term statistics. You're not sensitive to these regularities, definitely regularities that are not as commonly presented in dyslexia. One very last slide is what happens if you do the same experiment in autism. So we did the same experiment. It's not Sagi in this case, it's Shachar. We did the same experiment exactly with controls, again with the control population. Here again, the jump is here. The autism group is surprising. They never seem to reach the same level of response, even with 9 seconds interval. Which kind of, what's going on? Do they ever have a sufficiently large response? Maybe they simply respond less. So how can we decipher that? First, we can look at what happens in the very first trial. To their very first trial response, when they come to the lab, they haven't been exposed to these stimuli very recently, or as I said, it's pure tones, maybe never. Is it also smaller? I can say it's not smaller, but then how many responses per person do you have for the very first one? Just one. So obviously this is a very noisy response. We have the number of tries we have is just as the number of participants. It's not large enough. So the experiment we did is, suppose we have 30 seconds interval. Do people with autism increase their response if the interval is 30 seconds? I mean, but you cannot do a trial with this interval. Pom-pom, I won't let you wait for half a minute. It's pretty terrible. You cannot do that. It's really a startle effect rather than a trial. So we did it a passive experiment. We had a half a minute interval, and the guy who did this was Itamar. And Itamar, don't worry about this graph. It's just a control. The 9 seconds is a control. Basically, we look at whether there is a difference between the response in the first half of the block and the second half of the block when the interval is 30 seconds. This is a very large interval. We don't see a difference in the control group, average response first half of the block second, but in the autistic population, we see a highly significant adaptation even when the interval between every 2 tones is half a minute. Hugely long. I was so surprised. I thought that by 30 seconds we'll reach the same response. Verifying these results because it's so much longer than ever reported in the literature that I feel we need to verify that. So anyhow,
can I ask a question about this?
Yeah.
So it seemed before in the data you showed about the autistics that they really strongly underweighted the last trial, right? And here it seems that they're really weighting the last trial very strongly in a much longer delay.
So it's a good. Let me see if I understood your hinted question. If the last trial is 30 seconds away, will they incorporate it to a larger extent? That's the question?
So just if I'm understanding the graph we're seeing here, we're seeing that the last tone from 30 seconds ago actually impacted the autistics quite a bit, right? Even to a magnitude which is larger than the healthy subjects for a recent tone. So this in a way means that the last stimuli they heard is strongly incorporated as a prior, right?
It's a very good question. I can tell you what we have, which I didn't present here, and here we don't have, it's a passive experiment because so if you were to ask me what do I think happens, I think that with 30 seconds, had we had a behavioral effect, a behavioral study, I would see the effect in autism, but I would not see the effect in controls. We did ask participants to perform the task with up to 9 seconds interval, but then with 9 seconds interval, you see the effect in both groups. So with 2 second intervals, you see a larger effect in control. With 9 seconds, you see very similar effects. But I think that by 30, we would see, I'm not sure how to administer it because people fall asleep if you have such a long interval. And then you fall asleep, all sorts of things are different. So I would have liked to have this information, but I'm not sure how to acquire behavioral information with such long intervals. But it's much easier if you ask for what I predict. So I predict, and I may not be tested on it, that yeah, as you were suggesting, that I would see a contraction bias only in the autistic population and not in the control, right?
Okay.
It rings a bell with all sorts of other phenomena that people describe in autism that they might say after a while, oops, now I get it, okay, now I get it. But I can't really say in any direct way. Yeah, so let me just summarize. So in both dyslexia and autism, we attribute the difficulty in long-term skill acquisition by atypical, in a way, sensory and sensory motor statistical learning, implicit statistical learning in dyslexia, but we propose an opposite type of atypicalities. In dyslexia, faster decay of the memory trace of earlier stimuli, which yield reduced accumulation of categories in general. I don't think it's specific to language or even to other sounds, but I think it's language and linguistic skills that really rely on it very heavily. Recently, Ayelet has conducted a study on, do people with dyslexia have difficulty in becoming experts in face identification? And the answer seems to be yes. So how come? We don't know. It's not the intuition. I think that typically we're not really experts in faces, except those that are very, very, very familiar to us. And if you look at this type of learning, which is very specific and item specific, then this type of learning seems to be slower in dyslexia in a way that is not very specific. In autism, we think there is opposite dynamics, there's slower online updating, and we think that it is particularly relevant to social context and sensory motor responses. But when you look at the literature, that people denote, for example, for motor kind of difficulties. So people note that there's a difficulty in catching a ball, but there's no difficulty in throwing a ball, where you initiate the response and you don't have to online update your motor commands to unpredicted plans. So overall, I think it is very consistent with large characteristics in the literature. And even though we got to these results from modeling online and basic sensory and sensory motor tasks, when we talk about these results with clinicians, they say, oh, of course. So I could say, well, one could say it's a bit disappointing. On the other hand, I find it a great achievement if you can link sort of these lab context stuff with clinicians' observations. Thank you.
Thank you very much, Mara. That was very stimulating and convincing. We just have a couple of minutes left for questions. So maybe students have 1 or 2 questions to ask.
Yes, thank you. I have a short question. Thank you very much for this talk. It's really interesting. It seems for the people with autism that they kind of have trapped priors, or they rely more heavily on the full amount of experience. Do you think that providing feedback for recent stimuli that you presented, for example, feedback if they perceived it wrong or something, like in your test, obviously it's not applicable, but in other tests, would you think it will increase their weights for their recent weights?
I don't have any direct data about it, but I would think not. I would think that the crux is in elongating the intervals, allowing for integrating this information. So a follow-up study that we're in the midst of is trying to map this, because it brings up the question in the general population, what is this updating mechanism? So, and how is it implemented in the brain? And I think, but this is really an hypothesis that I cannot strongly support, that it has to do with the cerebellum that is an updater. And the question is, suppose this is the case, what's atypical in the pattern or in the temporal pattern of cerebellar input into the cortex? And this is something we are collaborating now with Yifat Put, who studies this question with the task with monkeys. And she has a technique for temporarily eliminating the cerebellar input to the cortex and looking at how it affects both the performance, both the motor performance, and the pattern of responses. How, and getting back to your question, how feedback affects cerebellar responses, I mapped your question to my hypothesis, and I don't have an answer, but this is something one could test.
Great, thank you.
I have a little question as well, if I may.
Yes.
Hi, Merav. Thank you very much for your talk. It was really interesting. Well, I have a question which may be basic, but I don't have any prior knowledge on neuroscience because my major was not in that. But I was just wondering, because I watched one of your videos on your lab webpage, and you showed there 2 different sounds. And when, I mean, each person perceived it different. And when you showed a pattern in that sound, I mean, it's related to these 2 sounds that you said. You talked about it as well here, but when you showed a different pattern, then we heard either the sound went down or up, right, each time. And I was wondering, because I just learned this in class last week about the auditory system and all the basilar membrane. And I was wondering, because until now, I thought that it was very, very straightforward what happens in the brain. I mean, you get the sound and then the basilar membrane, all the mechanism and the encoding in the brain is, I thought it was straightforward. So do you know how to explain what happens once we have this perception and we have this skill acquisition change, and what happens in for example, in autistic people that is different from us?
So I don't think that the context modifies the basilar membrane. Interestingly, in the auditory system, there are feedback connections that go all the way back to the inner ear. I think that we're talking about higher level phenomena, but in the auditory modality, it's very, very densely feed-forward and feedback connections very densely interact. I don't think anybody can say where exactly is the source, unless you're talking about a very, very brief period from stimulus onset. But it takes us some time till perception develops. Sometimes I mean about 100 milliseconds, something like that. By then, all the auditory system had been activated. So if you look at the delay from the onset of the sound So the first response in the cortex, in the primary auditory cortex, it's less than 20 milliseconds. So by 100 milliseconds, everything has interacted. It's very difficult to tease it apart. And I don't think that we are at this stage yet.
Thank you.
Thanks, Mara. Just before we let you go, maybe one quick question. So I like it that you focused on statistical learning, but I wonder if what we're seeing here is a broader issue, broader deficits in memory, or at least in implicit memory, both in dyslexia and autism, that it's also manifested in statistical learning, but possibly also in other types of learning. So what do you think about the specificity of what you're finding here?
You mean, do I think that dyslexics also have a deficit in explicit memory?
No, but within implicit memory, it can be statistical learning, but it could just be like priming or things that are less statistical and more memory proper. And I wonder if they have a more general memory issue than unique to statistical learning.
So I will rephrase the question. The term statistical learning, I would say, includes everything. Now the question type, I think the question is the type of statistical learning. So in a way, the term has been snapped for historical reasons, by your ability to repeat sequences. So it's kind of how good we are in sequencing events. And I don't think that people with dyslexia have a difficulty in sequencing. This is something that we have asked. People with dyslexia have difficulties in other aspects of statistical learning. One of them, for example, I didn't show it, is gaining from zero-order statistics, which is the frequency of different items. They benefit less from high frequency of syllables. I don't think they have a difficulty in sequencing syllables. There are other aspects of statistical learning, and I think this line of studies is in infancy, because so much had been directed to our ability to remember the conditional probability between A and B. And I don't think that this type of statistics plays an important role in language learning, and I don't think it is impaired in dyslexia.
Okay.
Why do I not think it plays a role in language learning? Because language, the whole idea of being generative is not repeating the sequences you have been exposed to. For like, I will give you examples of morphology, roots in Hebrew, right? We have roots in Hebrew. For my elder daughter, she would say at the age of 3, "את יכולה להזכיר אותי." Obviously, you can understand what she meant, that I will do it for her, but she never heard it, and she never heard this combination. She took the roots. The whole idea is to take the roots and take them from one context to the other, rather than sequence them in the repeated sequence. If you use a repeated sequence, it typically means you didn't get the language. When I was a kid, I remember all the news began with, "הרי החדשות בעיקרן תחילה." I think I was 20 by the age that I understood what עיקרן תחילה means. I understood that this is something you say in the beginning of the news, and you don't say it anywhere else, and it says something about something important is about to happen, but I could not pass it into. So this is something, there is a phrase that is associated with a context. And maybe but this is not language. This is exactly what we don't think about language. And we have used this paradigm with many paradigms, many is two paradigms to test sequencing abilities in dyslexia, and they come out adequate. So if I say bada gasha, they would not be good with the bada gasha, but not with the ability to put them in the right order. And this sequencing is typically what people call statistical learning. So.
Okay, fair enough. Okay, thank you very much, Marav. Really appreciate it. And we had fun. Take care.
See you in person.
Hopefully soon.
It's very difficult to see what's in between waves, right? We thought that this period would be after the 4th, but not as at the onset of the 5th. But So have a lovely 2022.
You too.
Bye-bye.
Bye-bye.